I'd like to send you greetings on behalf of the Royal College of Pathologists UK, which I also represent. This has been a wonderful time and I learned quite a lot from the earlier speakers, and it's also wonderful because of a chance to meet up and catch up with a lot of old friends, both among the speakers as well as the participants. I must say that this is a rather interesting topic for me, and I feel a bit inadequate to talk about molecular pathology because if those of you who know me, my background, I started as a forensic pathologist who then switched my career to be a histopathologist with an interest in hematopathology. So I'm not a molecular biologist, not a molecular pathologist. I'm going to speak to you about molecular pathology only as a user of the service, of the technique, rather than a driver of the technology. So I've been asked to talk about molecular pathology and in terms of its impact upon lymphoma classification. As we know, we live in so-called the molecular age, where we hear a lot about molecular techniques. Now, the first question then, of course, is what are the molecular techniques are we talking about? I think if we consider immunohistochemistry and H&E stain as the standard assays, and then by ancillary investigations, we'll be thinking about in situ hybridization, fluorescence in situ hybridization, PCR-based techniques, and I'm going to focus a little bit about these and about how they may have impact upon the way we make diagnosis. First, in situ hybridization. Here, I think that in terms of lymphoma diagnosis, in situ hybridization to detect the presence of EBER as well as kappa and lambda are particularly important in the lymphoma diagnosis. So as you know, there are certain diseases such as NKT cell lymphomas, EBV-associated DLBCL. By definition, they are defined by the presence of EBV infection. And if you just do immunohistochemistry for LMP1, for example, you will not be able to detect the EBV because of the latency states. So in NKT cell lymphoma, you will find that the protein itself, LMP1, may not be expressed in some cases of NKT cell lymphoma because although the protein level tends to be at a latency state 1, and therefore you may not be able to detect it, and so you must look for the RNA. And I'm going to show you a little bit more about the use of some of these techniques. The other area that I think that will be of particular use in today's practice of lymphoma pathology is that of clonality analysis, looking at light chain and heavy chain rearrangement analysis, looking at T-cell receptor beta and gamma for clonality analysis. And I'll give an example of that. So this, for those of us who are not particularly familiar with molecular techniques, this is in situ hybridization. You have an RNA target looking for EBER or kappa and lambda, and you have a probe which targets the particular RNA target, and this probe is linked with a reporter molecule which can be alkaline phosphatase, which gives you a nice blue color, or you can convert that into a brown color with HRP and give you a picture that's more familiar to your immunostains. So this technique is now you're able to automate it. I realize I put my slides backward. So I'm going to talk today in terms of my scope, first about the ancillary techniques which are needed for lymphoma classification, and secondly, to what extent are the molecular tests required for classification in the current updated WHO 2016 classification. So as I mentioned about in situ hybridization, in the past we used to do that manually. Now we have machines that are able to do in situ hybridization for us, and you can either choose a blue color or you can choose a brown color depending on the machines you like. Some number of my pathologist colleagues like to have the brown color, so because it looks a bit more like the immunostains, whereas with the blue color sometimes they say I see a big blue drop. I'm not quite sure whether that is real or just a background. So it's up to your choice, but we have used both. I must say that I prefer to use a brown staining because sometimes I can combine the in situ hybridization with brown with immunostaining in red, and sometimes you can see that gives you a bit more information about which cells are the ones that are EBER positive. Is it a B cell like we see in the lymphomatoid granulomatosis, or is it a T cell as you might expect in NKT cell lymphoma? So sometimes that kind of a double staining technique may be of help. To give you an idea of to what extent do we really need these techniques, such as in situ hybridization and clonality analysis? Let me share with you this case. This is a case of a tumor from the oral location, oral cavity, in a relatively young person without much history of note. And what you can see here is a lymphomatous infiltrate composed of medium to large cells. Some of these large cells have big prominent central nuclei associated with that. So just looking at the morphology, you might say that this looks like some kind of a large cell lymphoma, looks aggressive in nature, some kind of high-grade lymphoma perhaps, but what is it? Okay, so we start off by looking at the immunophenotype, isn't it? So we'll look at the B markers, CD20, CD79A, PAX5, all negative. So maybe it's a B cell that's undergoing plasmablastic differentiation. So we'll try CD138, and this also looks negative. So maybe it's not a B cell neoplasm, maybe it's a T cell neoplasm. So let's try. So we see CD2, CD3, CD5, CD7. Now the only one that's standing up very nicely is CD3. Maybe we're dealing with a T cell neoplasm. And because, we'll look at, if it's a T cell neoplasm, look at some of the T markers. Well, they don't seem to express the T cell receptor beta or gamma, but there is expression, at least focally, of CD8, and there's a bit of granzyme B there. So maybe it's a cytotoxic T cell phenotype, and in our part of the world, maybe it's an NK T cell neoplasm. So we look at EBER. EBER is nicely positive, although CD56 is negative. Sometimes it can be negative in some cases. So maybe it's an NK T cell neoplasm. So maybe it's an NK T cell lymphoma. Well, actually, this is kappa and lambda. This is actually shows lambda light chain restriction by in situ hybridization, and you can see by PCR this actually is a B cell neoplasm. This is actually a case of plasmablastic lymphoma. And sometimes plasmablastic lymphomas don't express plasma cell markers such as CD138 or CD79A, as you can see here. And sometimes these cases can be difficult because they can show aberrant phenotype. So you can have aberrant expression of CD3 cytotoxic granules, and really can mimic other conditions. So these are difficult cases, and sometimes with difficult cases, we do need access to some of these ancillary molecular techniques, such as in situ hybridization, as you can see here with EBER, and also with PCR. So we don't need to rely on molecular techniques all the time, but there are difficult cases like this which require us to make use of some of these techniques. And here, I think in situ hybridization and PCR clonality are particularly useful from time to time. So then, from ISH for RNA probes or ribro probes for in situ hybridization for clonality analysis, which I've illustrated earlier on, what about FISH? Fluorescence in situ hybridization, of course, is a technique that is used to detect amplifications, deletions, translocations in particular. In the case of hematolymphoid neoplasms, translocations are best detected by FISH, as we well know that because of the difficulty in the binding of the primers in the case of follicular lymphomas, this is much more sensitive than PCR. FISH is not difficult to do. The idea and the principle is quite easy. The idea here is that you have a template which you denature by heating, and then you have a probe which targets the particular genetic alteration you are trying to determine, and it is labeled with a fluorescent dye, and that of course hybridizes against the target. And you can look for translocation two ways. First, you can use a break-apart probe. So in a break-apart probe, you know where's the break point. You designed the probes, one colored green, one colored red on either side of the break point. So normally, if there's no translocation, red and green are together. If there's a translocation, it's broken apart, the red and green get separated. Or you can use a dual fusion probe. Here, the idea is that you must know which are the translocation partners. So one gene is you colored red, one gene is colored red. Normally, the red and green are different because they are on two different chromosomes. They are far away. But if there's a translocation, the red and green come together, you get two fused signals like this. So this is a simple way of detecting translocations using either a break-apart or a dual fusion probe. Now, for many cases that are characterized by translocations, mantle cell lymphoma, follicular lymphoma, We know that they have the translocations which define a large proportion of these diseases. But do we really need fish to make the diagnosis? Well, usually not. You've got a nice mantle cell lymphoma with CD5 positive, 23 negative, cyclin D1 wall-to-wall. Why do you need to fish? You can make the diagnosis of mantle cell lymphoma. Similarly, you've got nice follicular structures in a lymphoma that has got CD10 positivity, BCL2 overexpression in those neoplastic germinal centers. You don't need fish, again, to make the diagnosis. However, there are some difficult cases from time to time where fish may be helpful. Now, this is a case which was sent to me from a colleague, and the patient had a retroperitoneal biopsy. And it's a core biopsy in a retroperitoneal, and you can see that, well, I'm not whether sure you can see that there are follicular structures. So some people can see it and some people cannot. So there are those who can say that, hey, I think there is a little follicular structure here, whereas others will say you're just imagining things. Just look at diffuse infiltrate. And this is like kind of the follicular structure that's high power, but it's still a bit vague. And even a higher power, I can say that I think maybe there are some central blasts and central sites there. Another person will quite disagree with you. Well, maybe immunohistochemistry will help. So you do a CD20 and CD79A, you look like it's a B-cell lymphoma of some sort, especially when the Ki67 is rather low. But what kind of low-grade B-cell lymphoma is it? You don't quite see the nice follicular structures. If you look at CD3, there are a number of T cells there, but you don't, again, quite see the nice follicular structures. Although those of you who just believe there's a follicular structures may believe that there are follicular structures here, but others may not quite believe in it. Because in follicular lymphoma, remember that the neoplastic B cells will spill out into the interfollicular region. So there are a lot of cells here that are also there. That's also positive. Then perhaps BCL2 is positive. And of course, follicular lymphoma should be positive for CD10. The problem with CD10 staining here is not too clear whether perhaps imagine there might be a follicular structure here that's CD10 positive, but it's not very obvious. And so you're not quite sure. And some would say maybe those are granulocytes. Actually, I don't think so. There are some cells that are positive. If you look at BCL2, it's very difficult to interpret because there are a lot of T cells there, which are also BCL2 positive. So BCL2 is positive in the follicular structures in follicular lymphoma, but BCL2 is positive in all many B cells that are circulating. In marginal zone lymphoma, BCL2 is positive, mantle cell lymphoma, BCL2 is positive. So it may be difficult to interpret. CD23 is a bit more helpful, highlights the number of follicular structures that you can't quite see on the immunostains. In any case, the pathologist wasn't quite sure, especially when the CD5 also shows some staining here. That's because there's a lot of T cells, which of course are CD5 positive. So therefore, perform FISH on this case, and FISH is very easy. You can see that there is indeed the 14-18 translocation as you expect to see in follicular lymphoma. So sometimes with very small biopsies, where the features are not very typical, FISH can be very helpful in making these diagnoses. But most of the time, we don't need FISH to make diagnosis of follicular lymphoma or mantle cell lymphoma. Now, apart from FISH, the next step we talk about sequencing in terms as a molecular technique. To be honest, it's only more recently that we use sequencing for mutations in the lymphoma diagnosis, because most of the time in B-cell lymphomas you see translocations which you can detect by FISH, clonality analysis, PCRs, that's about all we do. But in more recent years, we know that there are a number of mutations that underlie various lymphomas, and therefore recognizing these mutations may be useful. So first, we talk about Sanger sequencing, the old favorite as originally described by Fred Sanger. The idea of, and the principle is quite simple. The idea here is that you have a template that's containing the sequence of interest. You have a primer just ahead of this, and then you put into the mix a number of the deoxyribonucleotides, ATCGs there. At the same time, you put a number of D-deoxynucleotides. And the idea of these deoxynucleotides is that they will stop the reaction. They will not allow the chain to progress. So after a few cycles, So after a few cycles, of PCR, what will happen, of course, is that you get different chains of different lengths, and because you have got there these dideoxynucleotides there, each of which carries with it a particular fluorophore, then of course you will stop the chain reaction at different stages, and so you got different chain lengths, and each one of this, of course, can give you a fluorophore, which you can then detect in this way when you send a laser through it. And through this, you can line the sequences by the length right through, and then read off the mutation, the nucleotide sequences. So this is the traditional Sanger sequencing, which is now automated, and you can determine, for example, in this case, looking for the MYD88 mutation, that this case has a particular mutation. So this is Sanger sequencing, and that is still done most of the time. The problem and the usefulness of this is that although this is a very useful and easy way of detecting mutations, it detects one mutation in the strand at one time. And so if you have got a mutation that is sort of very simple, a simple mutation such as the MYD88 mutation involving a single nucleotide switch, now Sanger sequencing does that very well. If you've got BRAF mutation, you can use Sanger sequencing, that detects it very well. But if you have got a very big gene that is like a BRCA mutation, then where is it the mutation occurs? There can be so many different mutations. Using Sanger mutation is going to be quite laborious. It's going to be costly as well. So you need a better way, and we'll talk about that later on. So currently we use Sanger sequencing for MYD88 mutation, which we see in, of course, lymphoplasmacytic lymphoma. You see that in some cases of DLBCL. We use the Sanger sequencing for BRAF mutation as well. But for these kind of mutations, with sort of a single small mutation, it's easy to detect this mutation using allele-specific PCR. And now what this does is that since you know that the type of the mutation is not a whole lot, it's just one particular kind of mutation, involves one nucleotide change, then what you do is that you design a primer that will only bind when the mutation is present. So if there's an A to C mutation, you design a primer that will bind when the mutation is present. So what will happen is that you can use a quantitative PCR technique. If there's binding if there's a mutation there, then the primer will bind to the mutation, and then during quantitative PCR, of course, you can see amplification. But if the mutation is not present and your primer will only bind to the mutated sequences, of course, nothing will happen, so you will get very little amplification. So this kind of a technique is very useful because it's very fast and it's an easy way of detecting mutations. So currently we use this technique to screen through large number of cases to detect the MYD88 mutation, which we then confirm using Sanger sequencing. So this is another technique that you can do. And there are a number of companies that provide various kits for doing this allele-specific PCR. Now, as I mentioned, if you have a single mutation, it's easy. You can use allele-specific PCR Sanger sequencing. But if you've got a very huge number of possible mutations in a gene, then of course that will not work. TET mutations, for example, looking at myeloproliferative disorders, myelodysplastic syndromes, the possible mutations are quite numerous. If you use Sanger sequencing, it will be laborious, it will be costly. So with that, we need to have a better way of detecting these mutations. Secondly, instead of detecting one mutation, suppose you want to look at multiple mutations. So as you know, that diffuse large B-cell lymphoma of the ABC type can signal through the B-cell receptor signaling pathway. You get mutations of CD79A, mutations of CD79B, you get mutations of the toll-like receptor pathway involving MYD88. So if you want to detect all these mutations using Sanger sequencing, it takes too long. So therefore, then perhaps we can use targeted next generation sequencing. And this is a technique that gives you the high sensitivity and low cost and high throughput. There are various different types of so-called next-gen sequencing, but the principle is about similar. The idea here is that, it's a bit similar to Sanger sequencing, but except that instead of sequencing one strand at a time, you take the sample DNA, you fragment it, make it into small little bits and pieces, and that's what they call constructing this library, the library generation. You break the sequences into small little fragments, and in each fragment, you tag both sides with a particular adapter molecule, so a particular tag. And that's the first step.
Then secondly, using this adapter molecule, you can amplify these fragments. So you get many different fragments of different sizes. And then using these fragments of different sizes, you can then line them up against a reference genome, and based on their overlying sequences, you can actually determine the sequences of the target, because you have sequenced each one of these in a massive fashion, sequence each one and then line them out all up. So this is what you do in the next-gen sequencing. These multiple fragmented sequences which are each one sequenced and then line them up. Can you imagine to line this all up properly will require a lot of bioinformatics, and there are many different bioinformatic pipeline analysis that will give you the analysis, and that becomes a bit of a problem, because depending on which kind of analysis software you use, you may get some differences in terms of the final results, which is a problem. And that is the problem with next-gen sequencing. Although, as you can see, there are plenty of these website and companies that will do next-gen sequencing for you to determine various mutations for you. One of the biggest problem, I have a different job in the Ministry of Health as a regulator, and one of the big problems is that how do we regulate a technique that potentially, despite being sensitive, in fact perhaps sometimes too sensitive, can give you different results depending on which lab you go to. The results might be different because the bioinformatics may be a bit different. And so at the present moment, as you know, the FDA has licensed only this one next-gen sequencing device for the detection of cystic fibrosis. And that is the idea that you're looking at one disease. You are looking at a disease using a standardized technique only with the MySeq platform, not with the HiSeq platform, standardized way of assessment. Then we can actually ensure a certain degree of reproducibility. So this is the major problem right now with the next-gen sequencing techniques. There is a lack of standardization, and so the risk is that a patient goes to one lab, and if there's a mutation found, go to a different lab, the read might not be quite the same. There is a movement coming on. There's an ISO 276. So this is a technical committee 276 with 5 different work groups, and one of the work groups is actually looking at the bioinformatics aspects. And the hope is that if we can come to some agreement in terms of how you standardize the analysis of next-gen sequencing, then targeted next-gen sequencing will be the way forward. Because in future, we will be able to look at multiple mutations at one go, rather than trying to sequence one at a time. So until that is done, in countries like Singapore, I understand Korea is a bit more advanced, they are already allowing that, but in Singapore we are still not using next-gen sequencing for clinical usage because of this problem about regulating it. So with that introduction to the various molecular techniques that may be of use in the lymphoma diagnosis and classification, as well as giving you some ideas of how we have been using some of these techniques. To what extent are these molecular tests required for the classification of lymphoma? There's a difference between the classification of lymphoma and that of prediction of response to therapy and prognostication. So I'm going to focus on classification. Well, if you look at the leukemias, the answer is quite straightforward. You cannot classify any leukemias nowadays without some molecular techniques. You already know that for the acute myeloid leukemias, but now with the B-ALLs as well, you can see that the classification depends on all these cytogenetic alterations. So without FISH, without classical karyotyping, it may be very difficult for us to classify these tumors. Then what about lymphomas, away from the leukemias? Well, the good news is that by and large, where lymphoma diagnosis is concerned, it still relies very much on a conglomerate of data from clinical features, from morphology, immunophenotype, as well as genetic alterations. However, quite a number of lymphomas are largely defined by their genetic alterations, and I'm going to mention some of them. Some of these cases, like follicular lymphoma, mantle cell lymphoma, although they are, the tumors are largely defined by these mutations, by these genetic alterations, you don't really need molecular techniques. You can detect them by morphology and IHC. So as you know, hairy cell leukemia, annexin A1 is very useful, sensitive, and specific marker can be used in many of these cases. Mantle cell lymphoma, we talk about cyclin D1. PLL, which involves the translocation of the 14-14 translocation or inversion involving TCL1, you can actually stain for TCL1 by immunohistochemistry quite nicely. So in the correct clinical setting and morphology, you have immunohistochemistry and H&E can actually help diagnosis without resorting to these molecular tests. I'm going to talk a little bit about some of these entities which are largely defined by the genetic alterations and to what extent do we need molecular testing. First, lymphoplasmacytic lymphoma. As we know, this is a disease of older patients presenting with cytopenia, splenomegaly, bone marrow involvement. They often have elevated IgM, which presents as Waldenstrom macroglobulinemia. The two are not the same. Waldenstrom is the clinical syndrome. The LPI is the underlying pathology behind it. But what has been discovered in the recent years, of course, is the finding of this particular mutation, the MYD88 mutation, in more than 90% of LPL. So therefore, this can be quite useful because we know as histopathologists, sometimes we struggle. Is this an LPL or is it a marginal zone lymphoma with plasmacytic differentiation? Or is it IgM myeloma? All these can sometimes produce IgM, and marginal zone lymphoma can sometimes IgG can sometimes be expressed in cases of Waldenstrom macroglobulinemia on one hand, whereas on the other hand, the marginal zone lymphoma with plasmacytic differentiation can be so florid that can mimic LPL. So with this finding of this mutation in more than 90% of LPL, you now have a marker to help diagnose this condition. This is, of course, an example of LPL in the bone marrow. You can see the lymphocytes, the plasma cells, many of which are stuffed full of immunoglobulins, which in this case, they are IgM positive. And you can see the lymphocytes staining up with CD20, the plasma cells with CD138, many of them, as I said, with immunoglobulin in the center, which you can identify as being of IgL monotypic immunoglobulin, so it's a clonal proliferation. And with this mutation, you can now be able not only to use this mutation to diagnose Waldenstrom, but also you can also use it to monitor patients of MGUS, which and also determine at one point in time when they're going to transform to significant Waldenstrom, the copy numbers actually will shoot up. And therefore, this also can be useful as a monitoring tool to monitor the response to therapy after treatment as well. So, very useful tool. We are now using sequencing for MYD88 for the diagnosis, the monitoring of Waldenstrom. And what did I do? Thankful. Okay, very scary. Okay, we can use allele-specific PCR and followed by standard sequencing, as I mentioned, and that's what we tend to do in the diagnosis and the monitoring of LPL. But now I'm going to talk about some diseases which are appearing in the updated 2016 WHO classification, which are defined by genetic alteration. And the first one I mentioned is this large B-cell lymphoma with RRF4 or MUM1 rearrangement. So this is a disease that is seen in younger people. So the median age is about 12, and they present with, quite similarly, with enlarged cervical lymph node or tonsil. So it's isolated, so it's a low-stage disease. And because they can present as both follicular, with a follicular morphology, diffuse morphology, or mixed follicular and diffuse morphology, they can actually overlap with the pediatric type of follicular lymphoma. Many of these cases were probably misdiagnosed as pediatric follicular lymphoma, and in the past we all probably considered all of them as pediatric follicular lymphoma. In the follicular form, as I said, it can be either diffuse or follicular, and in the purely follicular form, they have these large neoplastic follicles with quite similar to that of pediatric follicular lymphoma, except that in general they are not so serpentinous, not so geographic in our configuration. They tend not to have this starry sky appearance that you tend to see in the pediatric follicular lymphoma. Otherwise, they tend to look like large cells and therefore really can look like pediatric follicular lymphoma, which tend to be a high-grade morphology anyway. The important thing, of course, about this disorder is that they are defined by this rearrangement of RF4, which results in very strong MUM1 expression. This picture taken showing the similar appearance from the literature. You can see these follicular structures, which really in the past we probably might have called pediatric follicular lymphoma. And here you can see the very strong expression of MUM1. In terms of the immunophenotype, they are quite similar to follicular lymphoma because about 2/3 of them will express BCL6, 2/3 of them express BCL10, CD10 as well as BCL2. So it can really mimic follicular lymphoma. And of course, the characteristic feature about this disease is the rearrangement. Here's using a dual fusion probe. Here's using a break apart probe, showing you the rearrangements of MUM1 or RF4. So as I mentioned, the phenotype can overlap with that of follicular lymphoma with expression of CD10 BCL2 about 66% of cases. Most important feature is the strong MUM1 expression, and the only way to really define this is to demonstrate the translocation of RF4 to the immunoglobulin genes in a majority of cases. In terms of prognosis, this is very good prognosis like pediatric follicular lymphoma. But important to make a distinction because pediatric follicular lymphoma will not require chemotherapy, whereas this will require systemic chemotherapy, although the prognosis is good after that. So this is a new entity that's beginning to be recognized, which will require FISH for this recognition. Next, of course, I want to talk about Burkitt lymphoma. And Burkitt lymphoma we know very well is an old disease. You can see these starry sky appearance comprising of medium-sized lymphoid cells, very uniform in appearance. Very little no big prominent nucleoli as you can see, and this is of course the typical immunophenotype of Burkitt lymphoma, being positive for CD10, negative for BCL2, wall-to-wall, 100% Ki67 staining. I tend to do these additional stains to make sure I'm not missing a ALL, L3 kind of appearance. As you know, ALL, L3 is really Burkitt leukemias. There's an overlap in the phenotype, so I tend to do a TBT, make sure there's not a blastoid variant of a mantle cell lymphoma with a cyclin T1. So in most cases, we can make the diagnosis of Burkitt lymphoma, although as we know that the disease is defined by the translocation of c-MYC to an immunoglobulin gene. Many places, they are not able to do FISH for c-MYC. You can stain for c-MYC. And you can see wall-to-wall nuclear expression for c-MYC. You can make the diagnosis in most cases. Whereas if you are able to perform FISH, obviously this showing the translocation or rearrangement of c-MYC and IgH is the characteristic feature. Now, if you perform FISH, you have to make sure that BCL2 and BCL6 are, of course, intact. Because if c-MYC is translocated to not just IgH, but there's also rearrangement of BCL2 or BCL6, it's not going to behave like Burkitt lymphoma. That's a double or triple hit lymphoma, which we'll talk about later on. Similarly, if c-MYC is translocated, but it's not translocated to an immunoglobulin gene, but translocated to a non-immunoglobulin gene, that is, again, not a Burkitt lymphoma. So, even with FISH, you have to keep in mind what probes you use, and of course there's a cost element there. So I know that many times we don't do FISH. The other thing about Burkitt lymphoma, however, is that if you use just a dual fusion probe, the 814 translocation probe, to recognize Burkitt lymphoma, you will see some cases where it is Burkitt lymphoma, but your FISH will be negative. The reason why it's negative is because the c-MYC is not translocated to the IgH gene, so therefore your 814 translocation is absent, but instead the c-MYC may be translocated to a light chain gene, like kappa or lambda. So this is the case where c-MYC is translocated, but you can see that there is no translocation to the IgH is intact, but instead c-MYC is translocated to the Ig lambda. So that is also